Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. Here, my group and I will present the result of our group works. It's about prokaryotic and eukaryotic. Before that, I will introduce the members of the group. First, Ananda Nicola. Second, Fariza Wiharistiani. Third, Hatika Rahma Ramadina. Fourth, Nining Aruni. 5. Nuraini Fauziah. 6. Nurfadila Putri Fitriani. 7. Putra Fernando Dwi Ahmada. And the last, Herenita Imelda Jayanti. In here, before we present about prokaryotic and eukaryotic, there are several topics in the prokaryotic and eukaryotic. The first topic will be presented by Nuraini.
Thank you, Fariza. Okay, let me to explain about my chapter in here. I'm gonna tell you about comparing prokaryotic and eukaryotic cells. Cells is the basic structural and function units of every organism. There are two different types. The first one is prokaryotic and eukaryotic. Organism of the domains bacteria and archaea consist of prokaryotic cells. They are protists, fungi, animals, and plants, all consist of eukaryotic cells. Cells. The word of eukaryotic, it means true nucleus. That word comes from Greek, and the word prokaryotic means before nucleus, reflecting the fact that prokaryotic cells evolved before eukaryotic cells. There are a major difference between prokaryotic and eukaryotic cells is the location of their DNA. All cells share certain basic features. They are all bounded by a cellular barrier called plasma membrane. Inside all cells is a semi-fluid, jelly-like substance called cytosol, in which subcellular components are suspended. All cells contain chromosomes, which carry genes in the form of DNA, and all cells have ribosomes, tiny complexes that make proteins according to instructions from genes. The difference between prokaryotic and eukaryotic, the first is in an eukaryotic cells, most of the DNA is an organelle called the nucleus, which is bounded by a double membrane. Second, in a prokaryotic cell, the DNA is concentrated in a region that is not membrane enclosed, called the nucleus. The third, in a eukaryotic cells, in the cytoplasm of eukaryotic cells, suspended in cytosol are a variety of organelles of specialized form and function. That is the disparity in structure complexity between the two types of cells. The fourth, in a prokaryotic cells, membrane-bonded structure are absent in prokaryotic cells. That is the disparity in structural complexity between the two types of cells. The fifth, eukaryotic cells are generally much larger than prokaryotic cells. Eukaryotic cells typically 10 to 100 millimeter in diameter. Size is generally a feature of cell structure that relates to function, the logistic of carrying out cellular metabolism set limit on cell size. At the lower limit, the small cells known are bacteria called mycoplasmas, which have diameters between 0.1 and 1.0 millimeter. Thank you.
The next part will be presented by Ananda Nicola.
Thank you, Fariza. A panoramic view of the eukaryotic cell. In addition to the plasma membrane at its outer surface, a eukaryotic cell has extensive elaborately arranged internal membranes that divide the cell into compartments, the organelle mentioned earlier. The cell compartments provide different local environments that support specific metabolic function. So, incompatible process can occur simultaneously in a single cell. The plasma membrane and organelle membranes also participate directly in the cell metabolism because many enzymes are built right into the membranes. The basic fabric of most biological membrane is a double layer of phospholipid and other lipids. Embedded in this lipid bilayer or attached to its surface are diverse proteins. However, each type of membrane has a unique composition of lipid and proteins suited to that membrane-specific function. For example, enzymes embedded in the membranes of the organelles called mitochondria function in cellular respiration. Because membranes are so fundamental to the organization of the cell.
The next topic will be presented by Renita and Fariza.
Okay, I will explain animal cell function. For the first, ribosome. The function of ribosome is where protein synthesis takes place. And second, Golgi apparatus. The function of the Golgi apparatus is to form vesicles or pockets for excretion, to form plasma membranes, to form lysosome, and to process protein. Third, lysosome. The function of lysosome is to enter macromolecules from the outside into the cell by means of endocytosis. Decays material using phagocytosis control intracellular degradation and destroy damaged organelles or autophagy. Number 4. Mitochondrion. Oxygen and glucose combine to form energy, which need to from metabolism in organelles. Mitochondria in their sole form are called mitochondria. Has the function of producing energy in the form of cellular respiration. Number 5. Peroxisome. The function of peroxisome is to reduce peroxidase from toxic metabolic bases and convert fats into carbohydrates. Number 6. Microvilli. Microvilli are added to group of cells known as villi. Microvilli play an important role in the absorption and secretory function of cell. The role of microvilli is to effectively increase the cell surface area. Number 7. Microfilament. The first function of microfilament is to provide assistance in cytokinesis, the process during which the cytoplasm of a cell divides into two daughter cells. The next function of microfilaments is to assist in cell motility and to allow organisms that have a single cell to move such as amoeba. Finally, microfilaments are also involved in the process of cytoplasm flow through the cell to supply nutrients. Number 8. Intermediate filaments. Intermediate filaments are also referred as intermediate filaments or intermediate filaments because they are located between the smaller microfilaments and the larger microtubules. Intermediate filaments in the cytoplasm function to maintain the shape of the cell, withstand pressure, and provide structural support to the cell. Number 9. Microtubules. It pushes the cell forward by forming a flagellata-like structure at its formation of cilia-like structure which can increase the surface area of the cell and allow the cell to move, help transport molecules or molecular materials. And finally, cytoskeleton. The function of cytoskeleton is very important, especially for cells that do not have a cell wall. For example, in animal cells, this type of cell does not get except for the thick outer layer. Thank you.
The next is centrosome. Centrosome is a region where the cell's microtubules are initiated. The contents of a centrosome is centrioles. Next is flagellum. Flagellum is a motility structure present in some animal cells composed of a cluster of microtubules within an extension of the plasma membranes. Next, plasma membranes. Plasma membranes is membrane enclosing the cell. The cell membrane separates the cell from the surrounding interstitial fluid. Cell membranes are made with basic ingredients in the form of lipoproteins, which are formed by fats and proteins. The next is endoplasmic reticulum. Endoplasmic reticulum is a network of membranous structures and tubes active in membrane synthesis and other synthetic and metabolic processes as rough and smooth regions. First, rough endoplasmic reticulum is a series of connected flattened sacs part of a continuous membrane organelle within the cytoplasm of eukaryotic cells that plays a central role in the synthesis of proteins. The rough endoplasmic reticulum is so named for the appearance of its outer surface, which is studded with protein synthesing particles known as ribosomes. Next, smooth endoplasmic reticulum is a meshwork of fine disc-like tubular membrane species, part of a continuous membrane organelle within the cytoplasm of eukaryotic cells that is involved in the synthesis and storage of lipids, include cholesterol and phospholipids, which are used in the production of new cellular membranes. The smooth endoplasmic reticulum is distinguished from the rough endoplasmic reticulum, the other basic type of endoplasmic reticulum, by its lack of ribosomes, which are protein synthesizing particles that can be found attached to the outer surface of the rough endoplasmic reticulum to give the membrane its rough appearance. Smooth endoplasmic reticulum occurs both in animal and in plant cells. The next is nucleus. There are three types of nuclei in the nucleus. First, nuclear envelope. Its double membranes include the nucleus, perforated by pores, continuous with endoplasmic reticulum. Next, nucleolus is non-membranous structure involved in production of ribosomes. A nucleus has one or more nucleoli. The last is chromatin, is material consisting of DNA and proteins visible in a dividend cells as individual coincident chromosome. The next part were represented by Latika and Putra.
Okay, thank you, Fariza. Now I will explain about cell plants. The organelles present in plant cells are first, nucleus. The nucleus is a highly specialized cell organelle. The nucleus stores the genetic components or chromosomes in a particular cell. The cell nucleus acts as the main administrative center of the cell, synthesizing metabolic processes such as cell growth, cell division, and protein synthesis. Next, plasma membrane. The plasma membrane is the part of the cell that is found on the inside of the cell wall. Functions of the plasma membrane are identification, cell communication, regulation of substance exchange, and isolation. Next, rough endoplasmic reticulum. Rough endoplasmic reticulum that is covered by ribosomes that appear speckled so that the surface is rough, is to isolate and carry these proteins to other parts of the cell. Next, smooth endoplasmic reticulum. Smooth endoplasmic reticulum is reticulum that is not attached to ribosome, so that its surface is smooth. Its purpose is to form fats and steroid. Next, central focal. Central focal is a large focal in plant cell. The function of the central focal is to store food reserves. The last, ribosomes. Ribosomes are small spherical structures that produce proteins. They are found in the cytoplasm or attached to the endoplasmic reticulum. Thank you.
Next, mitochondrion. Mitochondrion is powerhouse of the cell. It converts the energy stored in food, sugars, and fats into energy-rich molecules that cells can use. adenosine triphosphate ATP for short, in addition to serving as the site of cellular respiration in living things. Next, plasmodesmata. Plasmodesmata are small channels directly connect to system into neighboring plant cells. Next, chloroplast. A chloroplast is a plastid that contains chlorophyll and is used as a site for photosynthesis to take place. Chloroplasts are cell organelles that have a membrane found only in plant cells. Next, Golgi apparatus. Golgi apparatus are also called Golgi body. Golgi complex or diosome is an organelle associated with the excretory function of cells, and to modify protein by adding oligosaccharides to form lysosome. Next, peroxisome. Peroxisome are cytoplasmic organelles of plant cells. An important function of peroxisome is to assist chloroplasts in undergoing to the process of photorespiration. Next, cell wall. Cell wall is the rigid outermost layer of plant cells. It stiffens the cell, providing the cell with mechanical support and provide protection. Animal cells do not have a cell wall. Next, cytoskeleton. The cytoskeleton or cell skeleton is a network of protein bundles that make up the cytoplasm of eukaryotics. The function of the cytoskeleton is to provide mechanical strength to the cell, serve as the skeleton of the cell, and help the movement of the substances from one part of the cell to another. Thank you.
The last topic will be presented by Ninin and Nurfadila.
Thank you, Farida. Here I will explain about nucleus. The eukaryotic cells' genetic instructions are housed in the nucleus and carried out by the ribosomes. Genetic instructions are placed in the nucleus of eukaryotic cells and is done by ribosome. On the first stop of our detailed tour of the cells, let's look at two cellular components involved in the genetic control of the cells. The nucleus, which holds most of the cell's DNA. The nucleus contains most of the genes in eukaryotic cells located in the mitochondria and chloroplasts. The nucleus is the most conspicuous organelle in eukaryotic cells, average diameter of about 5 meters. The nucleus consists of a nuclear envelope to enclose the nucleus and separate its contents from the cytoplasma. The nucleus of the cell is bound by a double membrane called the nuclear envelope. This membrane separates the contents of the nucleus from the cytoplasma. The nuclear envelope consists of phospholipids that form a lipid bilayer. Laminae and nuclear matrix can help organize genetic material. Inside the nucleus, DNA is organized into separate unit scaled chromosomes. Each chromosome contains one DNA molecule bound to many proteins. The complex of DNA and protein that's making up chromosome is called chromatin. Each eukaryotic species has a unique number of chromosomes. For example, a typical human cell has 46 chromosomes in its nucleus. Thank you.
Okay, I want to explain about ribosome. We know if ribosomes are small and distant organelles found in cell and act as site for protein synthesis. Cells that have a high rate of protein synthesis must have a large number of ribosomes. An example is in human pancreatic cells, which have ribosomes in the million of them. Not surprisingly, cells active in protein synthesis also have prominent nuclei. If we look at the picture, The ribosome makes the protein into paste. At a certain time, the ribosomes are suspended in the cytosol while the ribosomes are bound, attached to the outside, the endoplasmic reticulum. Most the protein made in these free ribosomes function in the cytosol, while bound ribosomes generally make protein to be inserted into membrane as packaging for certain organelles such as lysosome or for export from the cell or namely its secretion. From the picture above we see that the ribosome has a very small size of 0.25 micrometer. And also on the diagram of the ribosome it can be seen that it has two subunits, namely the large and small subunit. The two subunits themselves consist of RNA strand and various kinds of protein. In addition to protein formation, ribosomes have other functions such as translation, which is the process of taking instructions from RNA and then turning them into protein. Then, in addition to its function for metabolic activity in the cell, so that is formed perfectly. Thank you.
Okay, everyone, it's end of our presentation. Thank you, and wassalamualaikum warahmatullahi wabarakatuh.